Alakazam. How do you interpret what a set of protein sequences given by the amino acid? What are some of their properties? Amino acids have a bunch of properties, but when you put them together in protein sequences, what properties do they end up having? And how do you calculate that? And what tools are available? Alakazam, bam, Alakazam is one of those tools. And that's what we're going to talk about here. Hi everyone, I am Saniya. This is cute little Yankee. And in this video, I want to talk to you about Alakazam. It's a really cool package available in R to help us calculate and figure out properties of protein sequences or protein probes. So if you have a set of amino acids, for a peptide or a protein, and you just want to figure out properties of them, how do you do that? And there are many packages available that I'll talk about. But in this video, I just want to talk to you about the Alakazam package. It sounds like an Aladdin kind of a name, Alakazam. So let's get started with this Alakazam package. But before I do that, Yangshi, I want to just review a little bit of the concepts involved with proteins and probes and sort of like what those amino acids exactly mean, just a little bit of a refresher. So please bear with me. So what we see is we see these proteins here. They have a bunch of different structures, they are composed of amino acids, and they have a primary structure, a secondary structure, and polypeptide chains as well. And then they have some sort of a structure so that, that's what this is showing here. And in the cell, what we can see is that, these proteins carry out almost everything. They're like their life itself. They give us life. They give us the ability to do all these complex functions and tasks and regulate breathing and, transcription, and all of these processes in our bodies are regulated by proteins, which is why it's important to also eat healthy, get all those important amino acids out there. And we have 20 different amino acids. So these amino acids, they're coming from messenger RNA, that is being interpreted by this set of alphabets. So I'm going to go through that right now. But basically what goes and constructs this is the job of the ribosomes in the cell. So if you look here, you will see that we have this animal cell. And this animal cell has ribosomes here. These help translate messenger RNA into polypeptide chains, sequences of amino acids. So basically, sequences of amino acids make up like polypeptide chains, and those could be proteins or part of proteins as well. But that's all done by the ribosome here. So the ribosomes are these small little dots here, small little circles as well. And they help kind of take this messenger RNA and help us kind of, come up with a sequence of amino acids, a polypeptide chain. And then that is ultimately, becomes part of a protein. So basically what we see here is that, again, this is the ribosome that I was trying to show you. These small little units, these small little dots, they seem so insignificant, but they're actually so important here. These small little circles in the cell, these are the ribosomes here. And the ribosome basically helps you read messenger RNA and helps recruit, these are transfer RNAs. They bring along the corresponding amino acid. They read each 3 bases at a time, and then they bring the corresponding amino acid. And then that forms a growing polypeptide chain here. So that's all done. This translation of this messenger RNA is all done by this ribosome. So, so these amino acids, they then form this peptide, and this peptide may itself form a protein or part of a protein. So, one messenger RNA, one polypeptide chain. So these polypeptide chains are a sequence of amino acids that are strung together. Again, with the ribosome sort of like the ribosome interpreting every 3 bases of the messenger RNA. So that's what we see here. These are sequence of amino acids. And the way that the ribosome, so the ribosome is very cute. The ribosome is saying basically, and you can see behind me just a set of like amino acids as well. Yeah, Yang. So a set of amino acids right behind me, but basically this little ribosome is saying, I need a threonine. Could you help me? So how we can interpret that is that this is the first letter, this is the second letter, and then within this are the 3 letters. So there are 64 combinations. So it's 4 times 4 times 4, where there are 4 different messenger RNA bases here. So instead of a T, messenger RNA has a U, uracil. So there's 4 options, A, U, G, and C, U, C, A, G. And basically 4 times 4 times 4, so there's 64 total combinations. Basically, there are 4 messenger RNA bases. So instead of having a T like DNA does, messenger RNA has a U for uracil. And the way that you interpret this, and I can have this in more videos, is that you just say, if you need a threonine, could you help? So basically the 4 bases, U, uracil, cytosine, adenine, and guanine for messenger RNA. So 4 for the first position, 4 for the second position, 4 for the third position. So this is 64. So there's 64 total codons, which are 3 amino acids long. And basically, like if you want a threonine, a THR, then there are 4 different possibilities. The first base is going to be an A. So in the first letter, we go to an A. Then the second one has to be a C. So all of these 4 have an A and a C in common. That's what we notice about all of them here. They have an A and they have a C here. So that means that you have to start at A and the second letter has to be a C. And then you can have all sorts of combinations here. You can have ACU, ACC, ACA, and ACG, and that will give you threonine. So all of these show us the 20 different amino acids that are out there. And all of these 20, they have their own code as well. They have properties about them. Like aspartic acid, as you'll see here, ASP, aspartic acid. Then there's threonine here, for instance. So threonine is T and it's uncharged and polar. So all of these amino acids, all of these 20 amino acids have a bunch of properties. They can be negatively charged, positively charged, uncharged and polar. All these are properties that they have. And again, this is just another way to see this amino acid dictionary here, you know, where if you want to see what ASN is, that means that for ASN, the way that you interpret it is that the first letter is an A. So these are the 2 options for ASN, but it means that the first letter needs to be an A, and the second letter also needs to be an A, and then for the third letter, it can be a U or a C, as shown by here. So it's kind of like a very cute dictionary. And you can see that all of these 20 amino acids, they have a 1-letter code. So when I will show you a bunch of the sequences right now, what you're going to observe in this situation is the following. So I'm going to show you how we now use this Alakazam package. So when we're working with them, we're going to be working with these proteins, with this sequence of amino acids over here. And again, remember that all of these here, these amino acids, they have values, they have letters that are used to represent them. ASN, glycine, you know, all of them have like some way to interpret them. You know, MET, which starts the, polypeptide chain, MET, that is given letter M. So that's sort of how we're going to be working with these. We're going to be working with them in terms of their 1-letter code. You know, again, these are just 1-letter code. This is another way to see the alphabet as well. Threonine, you know, all of the P, and that's just basically how you can interpret all of these codes with 1-letter code. So I'm going to show you now how, if you do not have Alakazam, please install it using BioC Manager, install Alakazam. So I already have it there, but if you don't have it, please install it. So I'm going to just load in Alakazam right now. So I loaded in Alakazam. And now let's say that these are 7 protein sequences or polypeptide sequences here. And you can see that these all map to amino acids. So we can interpret the amino acids that they map to, for instance. You'll notice that all of these kind of map to amino acids over here. all of these letters, like this M is methionine as well. Then this E stands for GLU, which is glutamic acid. You know, that's what this E stands for here. So there's lots. And then this P here, for instance, stands for choline. So, and then we can look at choline and see what does PRO, what kinds of amino acids make up choline. So choline can come from CCU, anything with CC, U, C, A, or G can be the third base, but it's occupied all of these here. So basically, we have these protein sequences of various lengths, and we want to find properties about them as well. So again, each of these amino acids can have their own set of properties as well. They can be positive, negative charge, polar, you know, able to work with water, not able to work with water. They can have various properties, but we just want to figure out for these sequences here, what are their properties? So I have a set of a vector of these sequences here of polypeptide chains. And again, you can just think of them as kind of comprising, you know, amino acid sequences like this. So we want to just figure out what are their properties. So we're going to first put them into a data frame like this. So we have a data frame of these protein properties data frame here. There are nine sequences here, 9 unique protein sequences as well. So there are a bunch of different functions available in Alakazam. So the first one is polar. So what happens is you just take in a vector of protein sequences like this. So if you put them into this polar function, it will return the average polarity. So that means that for the first one, you know, we have this 7.706250 for this. Then for this other sequence, it looks like this value here. For the third sequence, it is this. This is the polarity values. And then you can plot the histogram here. And when you plot the histogram, all you do is you take the polarity vector, you can have your title here. You know, I have the color of the title that I want. I have the color of the bars. I have a subtitle, you know, the scale used is R. Grantham's amino acid difference formula to help explain protein evolution 1974. So I just had a few different parameters that I've used here, but this is the histogram. Now, maybe they correspond to a study or something from what the histogram looks for the average polarity scores. And I can see from the quantile, just the distribution as well, like the 0th percentile, the bottom value, the 100th, the maximum, the 75th percentile, 50th percentile, 25th percentile. That's what this tells me. I can also put it into a data frame. And similarly, I can also look at the gravy. No, the gravy is not for Thanksgiving. The gravy is not for Thanksgiving. Oh, he's hungry. No, the gravy is instead the grand average of hydrophobicity, like being scared of water. So I can run gravy for that too. I can run bulk, average bulkiness, aliphatic vector, you know, aliphatic index, and charge. And then what I can do is I can add this to this data frame. So from this data frame, I've been just trying to capture the protein properties. So one of the things is that for my protein sequences vector, I also want to capture the number of amino acids in them. So nchar allows me to calculate the number of amino acids. So this is 16. This is 16 long. This is 17. So basically, I'm just going to add this here. So this says that we have this data frame from before. I want to add in this amino acid length column, and it should just be the number of characters in each of these in the vector. So if I were to look at this right now, protein properties data frame, I see all the original like sequences that the 9 different sequences here and their corresponding respective amino acid length. This is really small. This is just 9 amino acids, but this is allow you to look at what are the number of amino acids here. And then I also we ran gravy from before, you know, grand average of hydrophobicity. These were the values for gravy. You know, so I can just add those here. This is for bulkiness. This is for aliphatic. This is for charge, and then this is for yeah, I think this is a repeat. Yep. So basically, now what we see is we see a bunch of properties. We see amino acid length, grand average of hydrophobicity gravy, average bulkiness, aliphatic index, net charge. So there are a lot of different values that we see out here. A lot of different values. And what we want to do is we can look at the quantiles for the gravy vector, for instance, see the distribution of the values for the grand average of hydrophobicity as well. So we can look at gravy, the grand average of hydrophobicity values. We can see how they are distributed for the 0th, 25th, 50th, 75th, and 100th percentile. We can do the same thing for the bulkiness, for the aliphatic index. That's what quantiles does, and for the charge as well. We can plot histograms as well. So what we can do is we can look at the histogram of the net peptide charges for the protein probe sequences, what those values look like. We can look at the histogram for the aliphatic indexes for these 9 proteins. If you count them, if you look at these values for aliphatic index, 2 plus 2 plus 1 plus 2 plus 2, this would be 9. But the charge vector, you would see that it has negative charges and positive charges, and then these all should add up to 1. We can also look at the histogram for the bulkiness score as well. We see a lot of them are centered around here, 3 out of the 9, a third are here. For the gravy, grand average of hydrophobicity scores for the protein probe sequences as well. So there are a lot of different metrics here. And I just want to sort of show you, so we talked about just these different functions that we used here. I was mainly showing you how, we can look at the polarity, and also how we can look at gravy, bulk, aliphatic, and charge as well. Oh, that's what I was missing. I forgot to add in this. I know I was missing something. I forgot to add in polar. And that's what this is, polarity vector. So I can just call it polarity. And then here I can just have this. Ultimately, these are my properties for these protein probe sequences, these amino acids. We have the polarity, net charge, aliphatic index, average bulkiness, grand average of hydrophobicity, and amino acid length as well. So there are more, but I just kind of wanted to show you that we use different packages like Alakazam here, and they have different functions like polar. It finds the average polarity score of amino acids. So non-informative positions are excluded. Any of these things, they do not consider them. They need a vector of strings consisting of amino acid sequences, which is what we supplied here. We supplied these protein sequences like this. You can have one or many, but it just needs this as input. And then it's going to try to calculate this for us. So we can even see, for instance, if they give us this default scale, polar seq, this will be 8.55 and 8 for these two respective amino acid sequences. And then there's gravy that calculates the grand average of hydrophobicity, gravy index of amino acid sequences using this method of kite and Doolittle. So again, they get rid of non-informative positions. They just kind of need these sequences to calculate these. Then there's bulk, which calculates the average bulkiness, how chunky amino acids are in the sequence, and they don't consider the non-informative positions. And then this is the references that they're using here, a vector of bulkiness scores for the sequences that you give. So again, they just need amino acid sequences like what I've shown. And then aliphatic, the aliphatic index of amino acid sequences using the method of Ikai. They don't consider, if you have characters in there, they don't consider these characters. If you have normalized as true, then it kind of divides the aliphatic index of each amino acid sequence by the number of informative positions. Lastly, the charge calculates a net charge of amino acids using the method of Moore, 1985. It does not include the C-terminus or the N-terminus charges. Environmental pH, you can specify that. So you need a vector strings defining amino acid sequences. You can specify environmental pH. If you don't, the default is 7.4. You can also have the pK values for each charged amino acid, where the names are the single letter amino acid character codes. Otherwise, it uses some embossed scale. So this is null. And normalize is false, but if it's true, then it divides the net charge by the number of informative positions. So it does not look at these positions here. Like if your sequence has any of these, it doesn't look at them. And it just skips those. And normalizing is just going to kind of adjust the score based on, it does not count these in. But if it's false, it returns the raw net charge. But normalizing kind of similarly just adjusts for the fact that you had these special characters that weren't considered. So yeah, this is just some general tools for how you can better analyze, a set of, and these are histogram. This is code for histogram. You just have to put the vector in for the histogram. I'll show you guys again how, you can sort of run these histograms here. But basically, these are kind of how you go and you have a vector of 14 sequences, amino acid sequences that correspond to a polypeptide chain, and how you can figure out all these properties about them. So I hope that this helped you and that the method was like, Alakazam. Oh, and of course, there's one more thing I wanted to add, which is that, you can also just run these directly for just one, like peptide sequence. For instance, you don't need to put them in a vector. You don't need a vector of the sequences like this, because before we were doing is we were calling polar on a set of sequences here, or gravy on them, or bulk, or aliphatic, or charge. We were running them on all of these things here, gravy, bulk, aliphatic, charge. And again, these were the functions we talked about. We talked about polar, and then you have the sequence inside it. It can be a vector or a sequence. We have polar, we have gravy. The same thing again. I'll just put it in quotes. We have bulk. We have aliphatic. And then we also have charge. So these were the functions that we talked about. Now you can have a vector like we were doing right now, but you can also have individual as well. You can have individual sequences as well. You don't need to be putting in a vector. You don't need a vector to be put in like this 14 sequences. You don't need a vector. You can have a vector. Like if you want to, you can call a vector here on these, like what we were doing. But even if you just want to find them for one, like for instance, like let's say we want to call it for just this one, we can definitely do that. We can do this. You see, we ran the functions and also charge, the net charge. We can definitely do that. We could also have instead also just kind of represented this one as we can also just represent it like this, which is what it is. Actually, the MEGA is the first one, and we're going to get the same results. You'll see this 0.38 and this one. It's the same thing for the gravy in the spirit of Thanksgiving. So yeah, we talked about these functions, and I hope that you can run them. So in the spirit of Harry Potter, I'm going to say Alakazam. So again, I showed you can run this on one peptide or multiple peptides as well. So I hope that you run it and it works for you. So I'm actually not a muggle. I'm actually a witch. I go to Hogwarts and I know Alakazam, figure out the properties of proteins. It's pretty magic, right? Actually, this is just me trying to be. I really love Harry Potter, but Alakazam is sort of cool. It's sort of like magic. If I can tell you, I can find all these properties of proteins. You're going to think it's pretty cool because proteins define so much of our lives and who we are as people. So I think that's pretty magical, actually. So anyways, thank you guys for watching. Hi! I hope that you had fun learning about the Alakazam package in R on Bioconductor with Yangshi and me. Hi, little guy! So Alakazam, unlock the power and understanding of your set of protein sequences. So this is sort of inspired. I'm a Gryffindor, I think, Harry Potter fan. So Alakazam reminds me of magic, hence I'm wearing this little snuggie here. So I hope that this was a helpful video, and again, you just need protein sequences or probes that are expressed as amino acids where amino acids are represented by their alphabetic letter. And then this package just takes any sequence of amino acids, a vector or even just one character in it as well, and it sort of helps find these different properties like net charge, polarity, grand average of hydrophobicity, all these things. So you can even just put it in directly and it will calculate it for you. So don't worry about that there. So I hope that this was helpful for you. And again, please let me know if you have any questions at all. So please let me know if you have any questions. I'm Saniya and this is cute little Yangshi. And we're so happy that you watched this with us. So please let me know. And if you found this helpful, then Yangshi is saying, please subscribe to our channel. And please reach out with any and all questions. I hope that this was helpful for you guys. Thank you and have a great day.